Alright, hello out there to whoever is listening. I'm Pearson, and myself, Drew, Patrick, and Macy are going to talk to you today about some trematode parasites and their association with waterfowl mortality in the upper Mississippi. So a quick outline of our presentation. I'm going to introduce the parasites that we were looking at, talk a little bit about their biology and their association with the waterfowl, and then introduce the questions that we were trying to answer with the data we had. And then I'll hand it over to Drew. He'll talk about some of the study methods we use, the data manipulation and statistical analysis. And then Patrick and Macy will talk about results we got from these methods and discuss the interpretations and conclusions we can draw from our results. So, since 2002, there's been a substantial increase in waterfowl mortality in the upper Mississippi due to trematode parasite infections, and this is likely because of the introduction of the invasive faucet snail that these parasites use as intermediate hosts in their life cycle. And a couple of risks associated with these parasites is that they can potentially have impacts on humans pathologically. Both their larval and adult forms can cause issues for humans, and it can also affect the ecology of the surrounding area. If you have a lot of waterfowl being killed off from these parasites, that can have some cascading effects on the ecology of the area and other species involved in that food web. So the three parasites that we were looking at, Spiridiotrema globulus, which is up in the top right here, Sciathocotyle bushiensis, which is this one here, and Leognemyus polyune up in the top left here. So these three parasites are trematodes. They're Digynian flatworms under the Platyhelminthes phylum, and all of them have been found in dead waterfowl in the upper Mississippi River. So they all have complex life cycles that are pretty similar to one another. They use a snail as a first and second intermediate host, and then waterfowl act as a definitive host most often. And the intermediate host is often the invasive faucet snail, Bithynia tentaculata. And so there's the faucet snail up here, and then here is just to kind of wrap your head around the impact these parasites have. These are just dead waterfowl that have accumulated here in the corner of this man-made structure in the river. And then, so humans can also serve as hosts for the adult flatworms, or their larvae can cause things like swimmer's itch if there's enough of them present. So here's just the summary of the life cycle again. So the adults are living in the gut of the waterfowl. They release eggs in the feces. The eggs hatch into a miracidium, which swims around and infects the first intermediate host snail, reproduces and makes cercaria larvae that exit the snail and try to find a second host. They can reinfect the same snail, or they can find a new snail. Once they're in the second host, they insist, become metacercaria. Then these metacercaria are ingested by a waterfowl, and the cycle continues once they develop into adults. So the database we had essentially was a summary of waterfowl mortality across about a decade, 2003 to 2013. It looked at the waterfowl species that were found to be killed and the parasites associated with their death. So some questions we wanted to try to answer with this data was there an increase in mortality over that period of time? Was there a certain species of waterfowl that were particularly susceptible to infection or were found more commonly than others? And then was there any difference in male and female mortality in the waterfowl that were collected? So was a male or female of the same species more likely to be killed by these parasites? So with that, I'll hand it over to Drew, and he'll start to talk about some of our methods.
Hello, I'm Drew Leesacker, and I'll be covering the methods for specimen collection, data manipulation, and finally data analysis. So, a brief note, we were not explicitly given the methodology of how waterfowl were collected, but we do know that they were collected on pools 7 and 8 in the upper Mississippi River between the years 2003 and 2013, and then the birds were dissected and the number of parasites per species of parasite were counted. And then this data, along with the species of each dead bird, the number of each species of parasitic trematode per bird, the species of trematode that caused death, the sex and age class of the bird, were all compiled into an Excel spreadsheet, which was the database that we used for our data manipulation and analysis. So in order to analyze the data, we had to set it up so that it could be used properly for analysis. This just meant that although we had a total of 158 birds collected across the decade, for some of the tests we couldn't use all of the birds, for the example that for sex and species comparison, mortality comparison, we couldn't use all of the American coots that were collected as some of them had unknown sex. All the tests could be run in SPSS and Excel, and for the first test, a linear regression was run to determine if there was any significant difference in mortality over time or not, whether or not there was a positive or negative trend. For that, we just looked at the total number of birds per species per year collected to create our data points, and then a trend line could be applied, and we could look to see if there was any trends or not. Using this linear regression, we should note that regressions are not generally used as models to predict the future or maybe unknown data in the past. For the test to compare means of mortality across species, a one-factor ANOVA was run in SPSS on all the species that had 3 or more birds collected across the 11 years of collection. Decided to exclude species with 2 or less individuals collected over the decade, as this was considered insignificant amounts of mortality. Also, it's an extremely small sample size. These species that we excluded were American widgeon, the sorrel rail, the American black duck, and the goldeneye. For the actual analysis, if we found any significant difference between the mean mortality of species, we could run a Tukey AHSD post hoc analysis in SPSS to determine where the significant differences lied. This would point out if there was a significant difference, for say, between the American coot and the mallard mean mortality. The one-way ANOVA would just tell us that there was a significant difference between one of the species. Finally, for our last test, we could use a 2-factor ANOVA in SPSS to figure out which of the 2 highest mortality species had any difference in the mean mortality between males and females within the species, and if there's any difference between males and females across species, say comparing the mean mortality of male American coots to the mean mortality of lesser scalps and figuring out if there's any significant difference there. There was no need for a post hoc analysis in this test, as none of our factors, sex and species, had more than 3 variables. If, say, we decided to analyze the 3 highest species of mortality, we would have to figure out a post hoc analysis test to figure out where the greatest difference in mean mortality across species lied. For this one, we did have to exclude some of the individuals from our sample size. There were a total of 54 lesser scalps and 49 American coots collected, but we could only use 44 of the American coots and 49 of the lesser scalps, as there were 2 American coots with unknown sex and 5 lesser scalps of unknown sex. And then once again, this was spanned across the 11 years, so we had the total number of males from 1 year, total number of females from that year for the 11 years to create our means. Patrick will go over our results and the discussion.
Thank you, Drew. Well, my name is Patrick Fitzgerald, and I'm going to be going over the results from the test that we ran and how we interpreted that data. And the first thing we're going to talk about is bird mortality per year, and as that is for every species of bird. For the linear regression, we failed to reject the null hypothesis that the number of dead birds does not change as the years go on. There is a low correlation as well, with an R-squared of 0.0203. This is not a model to predict future mortality, but it is slightly indicative of the trend that the average number of deaths per year, which is 14.36, in this area will continue to remain stable. So, what does this all mean? Well, with an R-squared value of 0.0203, there is very little to no trend in the data collected. Because this was not a lab-controlled study, there are many variables when it comes to sample sizes and how much data can actually be collected. Looking at the data, you would expect to see more of a roller coaster effect instead of a trend in one way or another. This is shown as our data and actually means that the relationship between host and parasite is stable. In the figure, you can see a trend of highs and lows bouncing across this regression line. In years where you see higher mortalities, you will have higher parasite counts, but because there's less hosts now, you'll see a decrease in parasite numbers, so in the following years, you will see a decrease in duck mortalities. And then, because it is on the opposite end of the highs and lows, you will then see an increase in parasite numbers, leading to an increase in duck mortalities. If you look at the line, it looks like it is almost trending downwards. We would need a larger sample size and across many different years, though, to accurately predict if this is happening or not. The next thing that we're going to look at is mortality between American coot, lesser scalp, and sex. For these, a two-factor ANOVA was run to figure out whether there is no difference between species and then species and sex. What I mean by that is if there is a difference in mortality between American coot, males and females, lesser scalps, males and females, and then if there is a difference between males and females of the two species. In our test, we failed to reject the null hypothesis that there is no significant difference between species and species/sex. We can reject the null hypothesis that there was no significant difference in means between male and female. This would indicate that males and females have different rates of mortality from parasites, with males having the higher rate. All three parasite species were found in American coot, with C. bushiensis having the highest number and L. palliune only being found in this species. With lesser scalp, S. globulus and C. bushiensis were both found in it, and the S. globulus were at the highest number. Next, Macy will be discussing with you guys the results of bird mortality by species.
The final analysis was done to determine if there were any significant differences in the mortality between the waterfowl species that had at least 3 dead birds collected over the 2003 to 2013 time period. This was done to see if there are any species that showed significant differences. A one-way ANOVA was run to compare the average mortality of each species. A total of 152 birds from 11 different waterfowl species ended up being considered. We rejected the null hypothesis that all species would have had the same mean mortality with high confidence, with a p-value of less than 1/10,000th. A post hoc analysis then showed that of all the species, they either fit into a high or low average mortality group, labeled as A and B in the figure, respectively. Within each group, there was no significant difference between any of the species within that group, and there were no outliers observed. It should also be noted that since the collection was based on migration patterns, this is not proof of the parasites preferring Group A over Group B, but rather a comparison of birds that did use those waterways during that time. So, based on the method of collection used, the two species in Group A had the highest number of samples collected in Pool 7 of the Upper Mississippi River water basin from 2003 to 2013. Through use of statistical analyses, we were able to reject the null hypothesis that every species of waterfowl would experience the same rate of mortality due to trematode infections. Our data was split into the two groups. Most of them had low mortality, as stated before, but the lesser scalp and American coot did have significantly higher rates. This is a strong indication that these two species are somehow more susceptible to infection by the trematodes, they don't tolerate intensities quite as high, or they utilize food sources there that have higher trematode contamination that led to quicker death than maybe some of the other species would in those same waterways. Most of the birds collected had Bushiensis infections, but while the scalp had relatively low intensities, every single coot collected had this parasitic infection to some degree, and some even had intensities that were 5 times higher than the scalp, despite the coot being around 25% smaller in mass than the scalp. Around 85% of the scalp had co-infections with globulus, with intensities of that species that range from 66 to 46,000. That's over a 700-fold difference. With numbers that various and intense, it was unexpected that none were found in coots. But this does demonstrate how trematodes show definitive host specificity, which was also observed with the polyune trematode species. But it showed it even more, because the coot were the only waterfowl species throughout this whole study to have that parasite. Around 40% of them were infected when they died. However, they were never the only parasite in there, which suggests that they may not be a significant cause of mortality in this species, and they might not even be considered. So, in conclusion, the study was conducted to analyze a database of waterfowl collected in pools 7 and 8 of the Upper Mississippi River from 2003 to 2013, with documented intensities of the 3 major trematode species that cause mortality in waterfowl in the area: C. bushiensis, S. globulus, and L. polyune. This large, very thorough collection has never been assessed and provided a lot of insight to the cause of waterfowl die-offs that happen in our backyard. The significance of determining these parasite-host interactions that can cause mortality affects not just the birds, but us humans as well. These die-offs have many secondary effects that include waterfowl death, of course, but also include people hunting in the area, fishing, and even tourism as people walk by the Mississippi River on a stroll downtown. We were able to determine through this analysis that the mortality trends remain constant over time. They're not going anywhere. That the males are significantly more affected than the females, which is something that would be really interesting to look further into. And that the trematodes cause highest mortalities in the lesser scalp and American coot waterfowl species. Further analysis of this database would further provide insight to the specificity of the definitive host that the trematodes are able to infect and the extent of the parasitic interactions that occur during a co-infection by different digenean species. A brief look at the American coot bushiensis infection intensity was when there was only the one bushiensis present versus the intensity when both the polyune and the bushiensis were present in a co-infection. It showed that the intensity of bushiensis decreased by half. This is something that would be very interesting to look deeper into to put together why the co-infection rates with species and sex would be great to determine what actions to be taken to save our waterfowl. So by taking the information that we've gained through this study, through our species and sex analysis, really honing into those specific interactions is something that could be very useful further on. So I hope you learned something new today, and I look forward to further discussion of the host-parasite interactions that concern all the beautiful waterfowl species that utilize this upper Mississippi River during their migrations and the competition that occurs within the waterfowl themselves. These are our references, and we have all the links to the pictures embedded in the slides for you guys.